Hello everyone. Today we will be discussing about an important topic, which is the basic of dermatology, that is the anatomy of the skin and the basic skin lesions. As we all know that the skin is the largest organ system of the body. It measures about 2.12 square meter and weighs about 4.2 kilograms. The appendages, namely the hair, the sebaceous glands, the eccrine glands, the nail, the mucous membrane, and everything included within the skin, along with it, forms a part of what is called as the integumentary system. The skin is basically divided into three layers: the epidermis, the dermis, and the subcutaneous tissue. The epidermis is the uppermost part of the skin, which consists of four layers: the stratum basale, the stratum spinosum, the stratum granulosum, and the stratum corneum. These four layers are named from below upwards, thereby the stratum corneum being the topmost layer of the skin, which consists mainly of the dead skin cells. The layer below these four layers of the epidermis is called the dermis, which is divided further into two areas: the papillary dermis, which is the closer area to the epidermal part, and the reticular dermis, which is the deeper part of the dermis. And below the dermis lies the subcutaneous tissue, which includes the fat and the connective tissues. So, describing the layers in detail, the epidermis, the lowermost layer of the epidermis, is the stratum basale, which is also known as the basal layer or the stratum germinativum or the germinative layer, because most of the mitosis goes on in this layer. The cells of this layer are cuboidal or columnar, with a large oval nucleus and a dense basophilic cytoplasm. And these layer of the stratum basale lies over the basement membrane. The basement membrane is the zone which separates the epidermis from the dermis. It consists of various factors which bind these cells to the basal layer. The stratum spinosum is the second layer from the bottom, also known as the spinous layer or the prickle cell layer. This layer is 5 to 10 layer of polygonal cells with delicate spinous processes called the desmosomes, which are connected with adjacent keratinocytes. The third layer from below upwards is the stratum granulosum, which is 1 to 3 layer of flattened diamond-shaped cells filled with coarse basophilic keratohyaline granules. These keratohyaline granules first of all appear in this stratum granulosum, and hence the name the granular layer. The topmost layer is the stratum corneum, which is flattened, multi-layered zone of anuclear eosinophilic corneocytes, which is the dead layer shed during the epidermal turnover. At some places of the body, such as the palm and the sole, lies another layer over the stratum corneum, and that is called the stratum lucidum, which is a clear layer found only on the palm and soles, and that's why these areas are thicker as compared to other areas. The time taken for a cell to pass from basal layer to the surface layer and then being shed off is called the epidermal turnover or the transit time, which is usually in a normal person 52 to 75 days. In some diseases such as psoriasis, this turnover or the transit time is reduced to almost 3 to 4 days, thereby causing piling up of the skin in the form of scales. There are some other cells found in the epidermis beside the keratinocytes. The keratinocytes are the basic structural elements of the epidermis. The melanocytes, the Langerhans cells, and the Merkel cells are few other cells which are also found interdispersed between the keratinocytes. the melanocytes being found only in the basal layer of the epidermis. The melanin is formed through mediation of tyrosinase and DOPA. The basic structure is phenylalanine, which is converted to tyrosine, which is converted to DOPA, then dopaquinone, and then dopaquinone is converted to two types of melanin, the pheomelanin and the eumelanin. This conversion of phenylalanine to melanin is controlled by genetic factors, the ultraviolet rays, and hormones such as a melanocyte stimulating hormone. Melanin imparts color to the skin and hair, as we all know. It protects the skin from ultraviolet radiations, and also it is a biochemical neutralizer for free oxygen radical derivatives. Now coming to the epidermal appendages. These epidermal appendages are invaginations of the epidermis into the dermis, and several structures sprout out from these appendages, being the hair follicles, the sebaceous glands, the sweat, and the sweat glands. The sweat glands further are divided into three types: the eccrine glands, the epocrine glands, and the apoecrine glands. The hair follicle is found over the entire surface of the body except the palm, soles, glans penis, the clitoris, labia minora, and the mucocutaneous junction. The distal parts of the finger and toes are also free from the hair follicles. Now, the type of the hair are divided into lanugo hair, which are fine, soft hair in the fetus, the vellus hair, which are fine, short, non-medulated hair over the most part of the body, and the terminal hair, which are long, coarse, medulated hair over the scalp, beard, body, depending upon the age, and gender. This is the anatomical longitudinal section of hair. So this consists of the hair bulb, which is at the bottom, the stem, the isthmus, and the infundibulum portion. This is the cross-sectional area, cross-sectional structure. The inner part being the medulla and the outer part being the cortex. So the hair follicle is divided into an outer root sheath and the inner root sheath, and the hair shaft. The hair shaft consists of the medulla, the innermost part, the cortex, and the cuticle. The inner root sheath, which is outside this hair shaft, consists of three layers: the cuticle, the Huxley's layer, and the Henle's layer. And then beyond this inner root sheath lies the outer root sheath. These three structures are encased in what is called as the hair bulb. The lower portion is the follicular papilla, where it is the germinative layer basically, which has a rich blood supply. And outside these follicular papilla lie the melanocytes. Now the growth of the hair is divided into cyclical phases. The active phase of growth is called the anagen phase, which is the phase of growth activity, and it lasts for almost 2 to 10 years. About 90% of hair at a particular time are in anagen phase. The next phase, as the hair passes from the activity, is the phase of transition and is known as catagen, which lasts for 1 to 3 weeks, and about 1% of overall hair at a point of time are in catagen. Then, when the cessation of the growth activity occurs, the hair goes into a resting phase, which is called as the telogen. This phase lasts for about 3 months, and about 10% of hair are in telogen at this point of time. Telogen hair are shed off, and the hair next anagen phase hair replaces it. Average loss of hair is 100 hair per day. The next appendage of the epidermis is the sebaceous gland, which are lipid-producing holocrine multilobulated glands. The various types of glands you must have read in the first MBBS, which are holocrine, ecrine, apocrine. So these sebaceous glands are holocrine lipid-producing glands. These arise from the hair follicle at the junction of the infundibulum and the isthmus. These are distributed all over the body except palms and soles. These are numerous, large, productive over the face and scalp, and these are under the controls of androgens, particularly after the maturity and puberty. So the sebaceous gland consists of lobules of epithelial cells that differentiate toward lipid-producing cells. Enlarged vacuolated cells are present at the center of the lobule, and these disintegrate into an amorphous mass which forms the sebum. The sebum mainly consists of triglycerides, wax esters, squalene, cholesterol esters, and cholesterol. Various dermatoses which are associated with the dysfunction of the sebaceous glands include acne, rosacea, seborrheic dermatitis. The sweat gland, depending upon what mode of secretion they are following, are divided into eccrine glands or apocrine glands, and the apocrine glands have the features something of both. Every sweat gland has two parts, the secretory coil and the duct. The eccrine sweat glands are present all over the body, except the lips, the external ear canal, and the labia minora. Most are concentrated in the palm, sole, and axilla. The secretory coil lies deep in the dermis that connects with the surface by a duct. The most important role of these sweat glands is thermoregulation. The sweating by eccrine glands is under the control of sympathetic cholinergic nerve fibers. The central system controlling the sweat lies in the preoptic hypothalamic sweat center, which controls temperature, emotions, hormones, and gustatory factor leading to increase in sweating by eccrine sweat glands. The apocrine sweat glands are present in the axilla, the nipples, the periumbilical skin, the perineum, and the genitalia. Modified apocrine glands are ceruminous glands which are present in the external ear canal which produce wax, the Mohs glands in the iris, and the mammary glands. The secretory coil lies just at the junction of dermis and the subcutis. The duct opens into the mid part of the hair follicle just above the entrance of the sebaceous glands. These glands are innervated by the adrenergic sympathetic nerve fibers. The vestigial sexual function and represent scent glands, which are in human are a vestigious part but are very active in the lower animals. Now, the layer below the epidermis is called the dermis, which constitutes 15 to 20% of the body weight. The upper layer of this dermis, very much close to the epidermis, is called the papillary dermis. And the layer below is a thick zone which extends from the base of the papillary dermis to the subcutaneous fat and is named as the reticular dermis. Upward projection of the dermis is called the dermal papillae, and these strongly interdigitate with the downward projections, the rete ridges of the epidermis to form the dermoepidermal junction. There are three components of the dermis: the cells, the fibers, and the ground substance. There is a difference in the thickness of the skin depending largely on the dermal thickness, with the palm and sole being the thickest zones of the skin, almost 1.5 millimeter thick, and the thinnest being the eyelids in the post-auricular region of almost 0.05 millimeter in thickness. The males have a thicker skin as compared to females. Children and elderly have thinner skin than adults. Now coming to dermoepidermal junction, which is also known as the basement membrane zone, which consists of basal lamina, the lamina lucida, the lamina densa, anchoring filaments, the anchoring fibrils, and the dermal microfibril bundles. It functions mainly as an attachment of the dermis to the epidermis. It provides, through its anchoring fibrils, the filaments, it provides a support to the epidermis. These anchoring fibrils and the filaments are those structures by which the keratinocytes are attached to the dermis. The other function of the dermoepidermal junction is the regulation of the permeability of nutrients and exogenous substances. It influences growth, differentiation, and migration of basal keratinocytes. Autoantibodies to proteins in the dermoepidermal junction are responsible for a disease called as bullous pemphigoid and several other diseases and inherited defects such as the epidermolysis bullosa. This area, the dermal area and the reticular dermis particularly, has rich network of two types of sensory nerves, the corpuscles or the mechanoreceptors and the free nerve endings or the nociceptors. The mechanoreceptors respond to light touch and they are called as the Merkel cells, the mesonar corpuscles, and those which respond to pressure are called the Pacinian corpuscles, and these are located in the deeper dermis or the subcutaneous tissue. The nociceptors for pain and itch is transmitted. This sensation is transmitted through naked fine free nerve endings located in the basal layer of the epidermis, close to the dermoepidermal junction. The temperature sensation is being felt and transmitted by the process and bulb, which detect cold, and the Ruffini end organs, which detect heat. The heat, cold, and the proprioception are the receptors for these sensation are also located in the superficial epidermis, superficial dermis. Adjacent dermatomes often overlap, mainly in the this factor is very important for the local anesthesia. When we are giving a local anesthesia for any procedural excisions, for moles, etc. So there are dermatomal overlap, and anesthesia in one part might lead to an anesthesia in the neighboring part as well. The blood and the lymphatic supply of the skin consists of extensive subdermal and dermal plexuses. The dermal plexus consists of two horizontal plexuses which are connected by vertical communicating vessels. The superficial horizontal plexus lies in the papillary dermis, and the deep horizontal plexus lies just above the subcutis, and this supplies to the sweat glands and the hair follicles. The cutaneous vasculature is very important in thermal regulation, and the cutaneous lymphatics parallel the blood supply. Now coming to the applied aspects, the basic skin lesions, what are they and how to differentiate between various skin lesions and how they are important in distinguishing between one disease and the other. The skin lesions are basically classified as primary, secondary, and the special lesions. The primary lesions are those lesions which arise de novo as such as they are seen at the point of time. These consist of macule, papule, nodule, plaque, vesicle, bulla, pustules, and cysts. The secondary skin lesions are those lesions which are the result of some changes over the primary skin lesions. And these consist of scales, crust, erosion, ulcer, excoriations, fissures, sinuses, the scars, lichenification, and atrophy. There are some special skin lesions which are seen in some of the particular skin diseases: wheal, burrow, comedones, milia, purpura, telangiectasia, sclerosis, poikiloderma, and target lesions. Now coming to the first primary lesion, which is the macule, and this is described as a circumscribed alteration in the color of the skin of any size or shape, which is non-palpable. This is a very important point. A macule is a non-palpable alteration in just the skin color. There is no depression, no elevation, no alteration in the skin texture as well. A macule which is more than 1 to 2 centimeter in diameter is called as a large macule. More than 2 centimeter in diameter is area. Earlier, any sort of macule was used to be called as a patch, but this terminology has been replaced now. The margins can be well defined or ill-defined, and it can be circular, oval, or of any irregular shape. It can be of different colors, such as erythematous, hyperpigmented, which is seen in melasma, and depigmented, which is total loss of skin color, and this is seen in vitiligo. So this is an example of hyperpigmented macular lesion, which is an example of melasma. This is a Becker's nevus. I'm sorry. This is a Becker's nevus. Again, when we go to palpate this lesion, this is just a visible color deformity. You cannot palpate it. You cannot differentiate it by palpating just from the neighboring skin. It is merged with the neighboring skin, but just the color differences help us to differentiate it from the normal skin. These are examples of macules. The next skin lesion is a papule. The papule basically are skin lesions which are raised and almost less than 1 centimeter in diameter. They might be of different colors. It might be brownish, as seen in Verruca vulgaris, a yellowish orange color, which is seen in xanthoma, violaceous or purple colored papules, which are seen in lichen planus, pearly white papular lesion, which is seen in molluscum contagiosum, or a malignant melanoma. The surface is rough or might be smooth. The other skin lesion is the nodule. A nodule is a solid, elevated, palpable lesion greater than 0.5 to 1 centimeter in the size, formed either by localized proliferation of tissue cells or infiltration with the inflammatory cells. The importance of nodule is that besides having a height and a width, it also has a depth, which differentiates this nodule from a papule. A nodule of more than 1 centimeter in size is better called as a large nodule. The shape can be anything from ellipsoid to globular or pedunculated or sessile. The surface can be smooth, keratotic, ulcerated, or fungating. The nodules, depending upon the depth of involvement, can be epidermal, dermal, a dermal, subdermal, or a subcutaneous nodule. So these are examples of nodule. These are example nodule of neurofibromatosis, and these are shiny, pearly, umbilicated papular lesions of molluscum contagiosum. This is a papule. You can see it is less than 1 centimeter in size, and it has just got a base and an elevated margin, a dorsal margin. It does not have any internal extension or a depth. The other lesion is a plaque. A plaque is a circumscribed elevated skin lesion which is more than 1 centimeter in diameter, and it is a shape of a plateau or a trapezoid which has flat base and slightly sloping or raised margins. So this is a well-defined erythematous scaly large plaque of psoriasis vulgaris. These are annular plaques because if you see the boundaries, these have two borders. One is the outer border and one is the inner border. Any structure which is circumscribed and having two borders is called an annular structure. So these are annular plaques seen in leprosy. A vesicle or a bulla is a fluid-filled lesion, depending upon the size, which is less than the size range has been changed now. It is almost 1 centimeter now. So an elevated fluid-filled structure, which is less than 1 centimeter, is a vesicle, and a bigger lesion is called as a bulla. The vesicle or bulla arises from the cleavage at either intraepidermal or a subepidermal level. In the bulla, the fluid in the cavity exerts an equal pressure in all directions, giving it a spherical shape. The contents can be either clear, a serous, or turbid or hemorrhagic structure in the fluid. Examples include vesicles of herpes simplex, bulla of pemphigus, scalds, and bullous pemphigoid. Any vesicle or any fluid-filled lesion which has a pus in the cavity is called as the pustule. The purulent exudate may contain either cellular debris, it contain leukocytes, it can be sterile, or may contain bacteria. They can be located at the opening of the hair follicles, known as the folliculitis, which you can appreciate in the diagram, in the photograph. And the pustules are superficial, they heal without any scarring because these are not deep, and the healing does not damage the internal structure leading to fibrosis. Cyst is a closed cavity that has an epithelial, endothelial, or a membranous lining and contains fluid or semi-solid material. The example consists of sebaceous cyst, the epidermal cyst, or a pilar cyst. The abscess is a localized accumulation of pus which is situated deep in the dermis or subcutis. Usually, it is not visible on the surface of the skin. It is erythematous, warm, tender, and a fluctuant nodule. Now, coming to the secondary skin lesions. What is an erosion? An erosion basically is a raw, moist lesion left from the complete or a partial loss of epidermis or a mucosal epithelium. It may result from trauma, a rupture of a vesicle or a bulla, or an epidermal necrosis. Erosions usually heal without any scar because it is very much superficial removal of the upper layer. And if at all it gets secondary infected, the infection might progress deeper and that might lead to scarring. Example of erosion are pemphigus. Whenever the bulla of the pemphigus ruptures, it leaves behind a superficial erosion. The Stevens-Johnson syndrome and the toxic epidermal necrolysis, when the sheet of the skin are removed, they leave behind raw erosions. An ulcer is a breach and destruction of the skin. It can be an epidermal or dermal or deeper enough, which can include the adnexal structures of the mucosa. Ulcers heal usually with scarring because these are deeper, maybe superficial or deep. The margin of the ulcers can be overhanging, such as in tuberculosis. It can be punched out, such as in gumma. The light pink granulation tissue at the floor is an indicator of healing in the ulcer. A fissure is a linear, triangular crack in the skin of the mucosa. It results from excessive tension or decreased elasticity of the involved tissue. It can be superficial or deep. The deep fissures are painful. The common fissures sites include the palm and sole because the skin layer over this is very thick and it cracks owing to pressure. A scar is the proliferation of the fibrous tissue that replaces the normal collagen after the ulcer involving the reticular dermis heals. The epidermis is thinned and wrinkled, and the adnexa-like hair are destroyed. So a scar does not have any hair follicle or sweat glands or even the sebaceous glands. Everything is destroyed because of the fibrous tissue replacing and um, uh, the entire tissue structure within the epidermis and if at all deeper. Hypertrophic scars are those scars which typically take the form of a plaque or nodule. It does not grow beyond the limit of the original trauma area. And the keloids are also actually fibrous proliferation and haphazard involvement of the collagen. But as the name described, keloid means claw-like projections. So the keloid extends beyond the skin surface of the trauma by the means of projection. So you can see several claw-like projection extending beyond the margin of the area of the trauma. Lichenification. Thickening of the skin due to repeated or prolonged rubbing or scratching of the skin leads to these three changes. First of all, the thickening of the epidermis and the dermis. The markings, if you see here, the skin markings are accentuated and the pigmentation is increased. So these three features, secondary to rubbing or scratching, that is thickening, increase in the skin markings and hyperpigmentation, is called lichenification. The examples of such diseases, lichen simplex chronicus, which you can see in this picture, lichenified chronic atopic dermatitis. The special lesions is erythema. Erythema is a local or diffuse redness of the skin which blanches on the pressure because there is a when we apply a pressure, the blood flow to that particular area ceases temporarily, and as we release it, this blood flow starts back. The main reason of erythema is vascular congestion or increase in the skin perfusion. Several diseases such as rosacea, viral exanthems, and systemic lupus erythematosus are associated with erythema of the skin. A comedone is a dilated pilosebaceous orifice which is plugged by keratin and sebum. An open comedone is the one where the pilosebaceous opening is open to the surface of the skin, and you can mark it very well by a black portion over the comedonal area. All these areas where we cannot see any black adherent portion over the top are called as closed comedones, whereas an open comedone, the sebum and all other debris are in direct contact with the air, thereby causing oxidation. And that results in a black plug form. Closed comedones are closed pilosebaceous follicle because the blockages is deep within. Milium. Milia is the singular form of the milium, and these are tiny, white, superficial cysts within the epidermal lining containing lamellated keratin. Sometimes arise on the blistered area as well when there is a damage to the deeper structures, such as in cases of dystrophic epidermolysis bullosa, porphyria, and dermabrasion. The most common site of these milia is cheeks. Erythema or purpura. What is the difference between these two things? An erythema, as we have just seen, it is due to the vascular congestion or an increased perfusion, such as in rosacea. Purpura are non-blanching, reddish to purple discoloration of the skin due to extravasation of RBCs in the dermis. This is an important factor. The RBCs, due to some cracks or due to some damages in the vessel wall, as is seen in some vasculitic disorders, the blood extravasates and deposits itself into the skin. So these lesions are non-blanchable. Petechiae are 1 to 2 millimeter small purpuric lesions, and ecchymosis are larger purpuric lesions which are formed by the extravasation of the blood. Diascopy, that means that when we apply a pressure over these areas by the means of a glass slide, we can see blanching in cases of erythema, whereas no blanching is seen in cases of purpura. Sclerosis is defined as a diffuse induration or thickening and hardening of the skin due to dermal fibrosis. The skin feels firm like board, immobile, because due to the fibrosis, severe fibrosis, the superficial layer of the skin is attached to the deeper layers. Example is morphea and systemic sclerosis. Poikiloderma is a combination of skin atrophy, telangiectasia, and pigmentary changes. This can be hypo or hyperpigmentation. Several diseases such as Rothman-Thompson syndrome, xeroderma pigmentosa are associated with poikiloderma. The beautiful target lesions, as you can see here, contains of 3 zones. The central zone, which is dusky, blistered or purpuric. The middle zone, which is a zone of edema and slightly pale, and the outer zone is an erythematous zone. These target lesions are characteristic of a disease called as erythema multiforme, which can occur as a result of viral infection, particularly herpes simplex virus, which is the most common cause, or it may also occur in reaction to certain drugs. The shape of each lesion determines what kind of disease it is. It helps us to differentiate between various diseases. It can be dome-shaped, flat-topped, umbilicated, acuminate, pedunculated, verrucous. How they are distributed, whether they are scattered, whether they are disseminated, whether they are symmetrical, asymmetrical, acral, confluent. The pattern of the lesions, annular, arcuate, linear, grouped, discoid, reticular, or gyrate, and the color of the skin lesions, which are purplish, which is very much specific to lichen planus, whether they are yellowish, xanthoma, etc. All these factors need to be taken care of while examining a skin lesion. First of all, whether it is primary, secondary, or a special one, what is the shape, so that we can come to a conclusion or a differential diagnosis. So this was all for that.